Hello guys, welcome to the channel. In this video we will be talking about the KREB cycle. It would be made super easy with very easy mnemonic guys, and what are the one liners that you need to remember for the MCQ aspect guys. Okay, starting with the video guys. See guys, first of all, KREB cycle guys, it is also known as guys tricarboxylic acid cycle. Okay, so this KREB cycle is known as the tricarboxylic. So basically what happens in the KREB cycle guys, it is the oxidation of acetyl coenzyme A guys to carbon dioxide and water guys. Okay, and this TCA is the central metabolic pathway guys. You see why we say so. You know that glycolysis produces the pyruvate, and this pyruvate is converted to acetyl CoA guys. Okay, and this acetyl CoA guys, okay, so this pyruvate is converted to acetyl CoA. Fatty acids can be converted to acetyl CoA. Ketogenic amino acids like leucine, lysine may be converted to acetyl CoA, and this acetyl CoA further goes to either to get oxidation to form either fatty acids, or to form hormone, or to form ketone body guys. Okay, so this is basically the central metabolic pathway that links everything. So this is the final common oxidative pathway guys, okay, for carbohydrate, fats and amino acid guys. Okay, so this is important guys. Okay, now see what are the reaction of citric acid cycle. Okay guys, see this is basically the table that we need to know for citric acid. So what is the mnemonic guys? How I have remembered guys. Okay, how I have remembered is guys, Allen career coaching, okay, institute, okay, of quota, sure success for medical officer guys. Okay, so basically I prepared for my NEET UG from Allen, so I remembered this mnemonic guys that time. So this is the mnemonic which I know, and I feel it's very easy guys. See, Allen, that is A for acetyl coenzyme A. C for career, that is citrate guys. Coaching, that is cis aconitase, that is the intermediate product guys, which is not stable guys. Okay, so it adds water and it converted to isocitrate. Allen career coaching institute. Off is oxalosuccinate. Quota, K is for guys. Sure success, S for succinyl coenzyme guys. Then success, that is succinate for medical student. F for guys fumarate, medical is malate, and officers is guys oxaloacetate guys. Okay, so this is all about the KREB cycle guys. So I think this makes it easy to remember you the KREB cycle guys. Now see, we need to know some of the things guys. Okay, which enzyme is inhibited by what guys? See, like fluoroacetate. Okay, it inhibits the enzyme aconitase guys. Okay, now talking about this arsenate guys, it inhibits alpha ketoglutarate dehydrogenase complex guys. Now talking about guys malonate. Okay, so this malonate guys, it inhibits the enzyme succinate dehydrogenase guys. Okay, so I'll give you a mnemonic to remember this also guys. Now see, what are the steps guys that involves the production of guys NADPH. Okay guys, see, if you see, there are 3 steps that involves the production of guys NADPH. See with me guys. Okay, first, okay guys, you see, then is the second step guys, and the third step, the one that I have marked in green and brown. Do you see guys? Now how do you remember guys? Remember, these are the main steps of the cycle guys. M means it is malate dehydrogenase guys. A means alpha ketoglutarate guys. Okay, alpha, MA. I means isocitrate dehydrogenase guys. Okay, then N is for NADH guys. Okay, NADH guys. So I think this makes it very easy to remember the steps that involve the production of guys NADH. And you know, 1 NADH is equal to 2.5 ATP guys. So we will have a calculation at the end guys. Okay, now see, coming on to what are the enzyme. When acetyl coenzyme is converted to citrate, see what is formed? Citrate. So the enzyme is citrate synthetase. Now coming on to the citrate, after citrate, Allen career coaching, that is cis aconitase guys. Okay, so what is the enzyme guys? Aconitase, because what is formed in the end guys? Aconitide, that is aconitase enzyme guys. Okay, now see, after forming of cis aconitase, we have isocitrate guys, because this cis aconitase is unstable, water is absorbed guys. Now after isocitrate, now you need to form oxalosuccinate, that is isosuccinate isocitrate dehydrogenase guys, that involves the production of NADH plus H plus guys. Now oxalosuccinate is formed guys. See, there is a release of one carbon here, guys, okay, and release of one carbon here, guys, okay. Do you see? So there is, so see, if you see acetyl coenzyme, guys, this is a 2 carbon compound, this, and this oxaloacetate, guys, is a 4 carbon compound. So the citrate that is formed, guys, is the citrate that is 6, remember, okay. This is a 6 carbon compound, guys, okay. Now this now if you see alpha ketoglutarate, guys, this is a 5 carbon. Why? Because their one carbon dioxide has been removed out from the reaction, guys. And if you see succinyl coenzyme, guys, that is 4 carbon compound, guys, okay. So this was about the ATP, okay. Now see, this is alpha ketoglutarate to succinyl alpha ketoglutarate dehydrogenase complex, guys, okay. Now see, succinyl coenzyme to succinate, that is succinate thiokinase, guys, okay. Now succinate to fumarate, guys, that is succinate dehydrogenase, guys, see. And here if you see, guys, okay, here it involves the, here you have FADH, guys, okay. It's not NADH, guys, this is FADH, guys, okay. So see, how can you remember, guys, this where the steps where fumes are formed, that is FADH wala step, guys, okay. Then from fumarate to malate you have fumarase, guys, and from malate to oxaloacetate you have malate dehydrogenase, guys, okay. So this was all about the cycle, guys, okay. And see, if you see, there you have a very special thing, guys, here if you see, if you notice, guys, okay, what is basically, this is basically a substrate level phosphorylation, guys, where a GDP is converted to a GTP, guys, okay. See, we'll talk about the energetics of the cycle also. This cis aconitase, guys, I said it is transient, guys, okay. So water is added, and what does it form? Allen Career Coaching, that is Allen Career Coaching Institute, that is, guys, it's form isocitrate, guys. Then isocitrate coaching institute of Kota. Do you see, guys, this is the oxidative decarboxylation. Why, guys? It involves the removal of CO2, guys, okay. This oxalosuccinate is also unstable, guys, so it undergoes spontaneous decarboxylation. And say, you can see this TCA cycle, guys, okay, this is both catabolic and anabolic. See, how do you remember what is catabolic and what is catabolic? Means katna, that is breaking down into smaller ones, and the anabolic is formation, guys, okay. So this is together is known as the, guys, this is together known as the amphibolic nature of the pathway, guys, okay, because substance enter the pathway and it can go further into other step of the pathway. So this was basically the mnemonic, guys, that will help you to remember Allen Career Coaching Institute of Kota, sure success for medical officers, guys, okay. See, so this was basically about the cycle what we have talked about. See, pyruvate, from here glycolysis form takes place, guys, and you have pyruvate, guys. This pyruvate is formed to acetyl coenzyme with the help of enzyme pyruvate dehydrogenase, guys, you see. Now see, revise just Allen Career Coaching Institute oxalosuccinate, they have not written because it is intermediate product. Ketoglutarate of Kota, sure success for medical officers, guys. And you see the steps that involve NADH. See, this step, guys, is the NADH wala step, this step is the NADH wala step, and this step is the NADH wala step. If you say fumarase wala step, okay, so basically there what is seen, guys, there FADH is involved, guys, okay. And this is a substrate level phosphorylation, guys, you see the substrate level phosphorylation, guys, that was this, okay. Now see, I have given the mnemonic. See, now remember, guys, this citric acid or the KREB cycle, guys, takes place in the mitochondria, guys, okay. I'll put out a video soon where I'll talk about all the biochemistry reaction, what is the site, guys, okay. See, now how to remember this malonate, guys, it inhibits the enzyme succinate dehydrogenase, guys, okay. See, this is the enzyme succinate dehydrogenase, guys, okay, and this is inhibited by malonate. So we know Mahendra Singh Dhoni, he is a great cricketer, guys. We know, like, all are great fan of him. So you can remember malonate, Mahendra Singh Dhoni, SD, that is succinate dehydrogenase, guys. Now see, alpha ketoglutarate dehydrogenase complex, guys, okay. So this alpha ketoglutarate dehydrogenase complex, this is inhibited by alpha A and A for arsenite, guys, okay. And this is non-competitively, guys. Remember, this keto diets, they are non-competitive, guys, okay. You know, keto is very good for weight loss, so this is non-competitive, guys. Aconitase is non-competitively inhibited by fluoroacetate, guys. See, in name only AC come, guys. Aconitase by fluoroacetate, guys, okay. See, so this was basically, I said, see, first we had the oxidative decarboxylation. Why? Because one carbon was lost. Again we had oxidative decarboxylation, guys, because one carbon was lost. This was a substrate level phosphorylation where GDP was formed 1 ATP, guys, okay. Now see, and we talked about other things in the cycle, guys, okay. So basically now talking about the energetics of the TCA cycle, guys. See, we had three main steps, MAIN, that is malonate dehydrogenase. 1 NADH enters, that is 2.5 ATP, guys. Isocitrate dehydrogenase alpha keto, again 1 NADH, that is 2.5 ATP, guys. Okay, now see, that is succinate thiokinase, guys, that is also known as succinyl-CoA synthetase, guys. Okay, that is a substrate level phosphorylation, so GDP will form 1 ATP, guys. Okay, then succinate dehydrogenase, guys, that was inhibited by, guys, malonate, guys. Okay, so that was basically FADH2, means it will form 1.5 ATP. So you can count the total number of ATP, guys, 2.5, 2.5, 16, 7.5 and 2.5, that is 10 ATP, guys. Okay, so basically 3 molecules of NADH and 1 of FADH2 are produced, guys. Okay, so this can come as a MCQ, guys. Okay, this is very important MCQ. See, we have already talked about the main source of ATP, guys. Okay, and is produced when main enzyme works. So we talked about all the enzyme, guys. Okay, FADH2 is produced when fumes are produced, guys. Okay, and succinate to fumarate step is mediated by succinate dehydrogenase, guys. You can see this step. Okay, and we also talked about the substrate level phosphorylation, guys. Okay, so that is by succinate thiokinase, also known as succinyl-CoA synthetase, guys. Okay, so I think hope this helps you guys remember the TCA cycle much easier, guys, and I hope you are able to solve the MCQ, guys. Do try solving the MCQ, do try with the energetics. Just remember the mnemonic: Allen Career Coaching Institute of Quota, sure success for medical officer, and remember the main mnemonic and the inhibition, guys, which is inhibited by, guys. Okay, if you like it, guys, please like, comment, share and subscribe, and do comment which topic you want the next, guys. Okay, thank you, guys. Happy learning, guys. Bye bye.